All right, guys, let's talk about lipids today. Lipids. All right, I like to start with elements first, so let's cover the elements and lipids. Elements and lipids, we have to have the one element that all organic molecules have, and that's carbon, right? Then the other two elements that we see in lipids are hydrogen and oxygen. Now, if you've watched the other video or listened to a lecture about carbs, carbs have the exact same three elements, but what's different between the carb and lipid elements is that these are in a 1-to-2 ratio, and there's very, very little oxygens available, which is going to kind of give us some pretty unique characteristics. Now, for a lipid, let's do the monomer. The monomer of a lipid is what we used to tell you is a fatty acid. That's not entirely the case. That's a part of it, but the other part of your monomer for a lipid is a glycerol. And really what we can do is we can start kind of building these things. Now, when we look at glycerol, if it has an OL, that means it's an alcohol. And look, if we have glyce up here, we've talked about that before. Glyce means sugar. So glycerol is literally a sugar alcohol. And what that looks like is I have C's. I have 3 C's with a hydroxyl group. Whoops. With a hydroxyl group. And then we have hydrogens off to the side. And then a fatty acid. All right, well, let's think about our functional groups. Acids, that's going to be my carboxyl, which is COOH. And then the fatty part, well, that's just going to be a hydrocarbon chain. And so what we want to do is we want to start building this monomer. So let's just look at one part of the glycerol, and then we can kind of add on. So if I look at one part of this, let's just do this right here. So carbon with a hydroxyl, and then I have the 3 hydrogens off to the side. And what I want to do is I want to add this to a carboxylate group. So here's my C, double bond O. There's my hydrogen, and then OH. And then stringing off of that is going to be a big hydrocarbon chain. And we kind of talked about this when we talked about organic structures. Every time I see something like this, I know that every point there's a carbon. And then off of those carbons, I have filling the rest of the 4 bonds with hydrogens. And so this right here, this is going to be a hydrocarbon chain. A hydrocarbon chain. All right, so now all I want to do is I want to build something. Every time we build, that means I'm going to do dehydration synthesis. And when I dehydrate, I'm going to take away water. Well, look, if I want to add this guy, this glycerol part of it, to this carboxylic with a hydrocarbon chain, I just have to look to see where the water comes from. So this carbon is going to break up. This carbon is going to break up. And then those two are going to join together. So what that looks like is I will have C, double bond O, OH. Actually, let's put the OH down here. OH, and then that's going to be attached to a big hydrocarbon chain. And then I just keep adding on to this sucker. So now I can do another glycerol. So here's my C, double bond O, and then a big, let's do it better, and then do a big hydrocarbon chain, and then I can do another one. There's that, C double bond O, and then a big hydrocarbon chain. So now that I look at it, this is actually something pretty familiar. How many chains do I have? 1, 2, 3. So I have 3, word root, tri, and because I added it onto a glycerol, this is a triglyceride. So triglycerides are usually the backbone to everything lipid-based. Because lipids, if we wanted to do some examples, these would be my fats and oils and waxes. But now that we look at the structure of this, that should make a little bit more sense, because what is the one thing that fats and oils and waxes all have in common is that they are hydrophobic. They're afraid. They don't mix with water. But if you look at the overall charges of everything, there's a lot of carbons and hydrogens in these fatty acids. Say we only have a few oxygens on this thing, which goes back up to my elements. I have lots of carbons, I have lots of hydrogens, and I have very few oxygens. And since I don't have oxygens, there's nothing for water to attract to, which will make it water-fearing, because there's nothing the water wants to attach to. So that's why oils and waters don't mix. Now, when we talk about lipid structure, we have a few different kinds of lipid structures. The first one, we've kind of heard of these words before. This is a saturated fat, or a saturated one. Now, without adding all the glycerols and stuff, a saturated lipid means the hydrocarbon chains are all single bonds. All single bonds. So it's just one straight hydrocarbon chain. Because they're straight, that means they can pack super tightly. And if they can pack super tightly, this means they are solid at room temperature. Because if I have a bunch of these saturated things, see, just like this, then they can pack super tight, and that makes it solid. So saturated fats, these are going to be like butter. See, the thing, butter is solid at room temperature. Waxes are separate, or are solid at room temperature. But if I do the next one, my next kind of lipid, I can have unsaturated. In an unsaturated, their hydrocarbon chains look really similar until you get to a double bond. And then when you get to a double bond, it kinks. So this double bond right here, a double bond, this is going to form a kink in the tail. And now that I have a kink in the tail, they don't lay flat anymore, okay? So double bond, right? Double bond right here forms a kink. That means they can't pack super tightly. And if they can't pack super tightly and they're just going to slide past each other, that means they are liquid at room temperature. And if I'm liquid at room temperature, think about what types of fats are liquid at room temperature. These would be my oils primarily. And the reason oils are liquid at room temperature is because somewhere in their hydrocarbon chains, they have a double bond, which makes them kink. Now, the last one, the one that's pretty bad for people, these are called trans fats. And what trans fats look like is they have that hydrocarbon chain, but now they have a double bond in there. Right there, they have a double bond there, but they don't kink. Trans fats are something that are man-made. There's not one natural trans fat in the entire earth or the entire universe that we know of. And since they're man-made, right, that means we don't have anything that can break that bond. And what's really, really bad about that is since they're a straight chain, that means they are solid at room temperature, which is really, really bad because if we think about foods that we eat with trans fats, remember, everything gets pushed through our circulatory system. So here's a really rough heart, and there's some arteries and veins going to and from. And so what happens is these trans fats, because they're solid and we don't have anything to break them apart, they start to build up in all of our arteries and veins because we don't have any way of getting rid of them. And then at some point, because they build up, there's not going to be any blood flow in or out of these. If there's no blood flow, this is where you're going to start getting massive cardiac arrest and heart attacks because there's zero blood flow going anymore. That's why trans fats are so very, very, very bad for people. Now, let's go ahead and do a couple special. So we have a few special lipids that don't really fit, that don't fit all the rules. The first special lipid is called a phospholipid. And if you've taken biology before, you've heard of phospholipid. Phos, that's referring to the phosphate. And the lipid, and the lipid is referring to that hydrocarbon chain. And so what that looks like is I have my PO4. So here's my PO4. And then I'm going to attach him to kind of a glycerol backbone. So there's my CH, and then there's CH2. No, not really a glycerol. He's just attached to kind of a carbon chain right there. And then coming off of those are a couple hydrocarbon chains. But here's the deal. One of them has a kink in it, and one does not. That means one of the fatty acid chains coming off is saturated, while the other one is unsaturated. Which, if you've taken biology before, and if not, we'll cover it in another video, every time we drew a phospholipid, we always made it look like a fancy person. Because here's their body, and then they have one straight leg and one fancy leg. You know how people always kick their knee up every time they're getting their picture taken. Well, the reason we drew them like this every time is because we're showing the saturated versus the unsaturated fatty acid tails, which we need to do a cell membrane because phospholipids, their entire function is they make up the majority, or let's do that. They make the majority of the cell membrane. And so every time we draw that cell membrane, every time we draw that circle, it's really just a bunch of these repeating themselves. And so it really looks something like this with a crooked tail and a crooked tail and a crooked tail, et cetera, et cetera. And of course, we'll talk about why later, but we actually have 2 layers of them, dot, 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 dot. But that makes a lot of sense because if my phospholipids, let's draw a pretty picture. If my phospholipids had 2 straight tails, well, think about characteristics of straight tails. Straight can pack. And so if I have a cell membrane that is solid, am I going to be able to get things in and out? No, I can't because it's solid. If I have a cell membrane that has liquid tails, they're both saturated. Well, if they're both liquid, is liquid going to be able to hold the shape of a cell membrane? They won't. They're just going to float away from each other because they fill the space. So we can't be like that. But the structure that works best is one with a straight or a saturated tail and then one with an unsaturated tail. Because this saturated tail, right, this is going to give it some type of structure, some type of form, and then the unsaturated tail is going to help with fluidity. So when I have both of those together and I draw my cell, that means he can hold the shape, but because some of it has liquid properties, I'm going to be able to get things in and out of it. And that's why these phospholipids, that's the best, that they have 2 of those tails, one saturated, one unsaturated. Now the other special lipid, these are my These are my steroids, right? Now, steroids primarily are going to be my hormones. And we can kind of talk about in the next unit why we want hormones to be lipid-based. Now, steroids always have the same structure. They have 3 6-ringed carbons. So I have 2, and then this one, of course, doesn't fit the norm. He goes up on this side, and then I have a 5 carbon. So steroids all have this basic shape right here, right? Which still satisfies all the elements because I have carbons at every point. Right there, and then I have the hydrogens attached. Now, what's different about them is off of this carbon right here, and generally off of this one, I can start having functional groups. I can start having other things attached. So sometimes that might have a different element. It depends on the hormone. But because it's overwhelmingly carbons and hydrogens, this is where we get to classify it as a steroid. Which, again, we'll talk about why hormones, etc., etc. Now, the last little bit, the thing we haven't covered yet, is just what lipids do, the function of lipids. The first thing is that they are able to store lots of energy, and they can store lots of energy in those carbon bonds, okay? Say right there, they store lots of energy there because remember every time we break a bond, we like to look for hydroxyl groups. Well, there's no hydroxyl groups there. So it takes a lot of energy to crack those apart. Because it takes a lot of energy to crack those apart, I actually get a lot of energy in return, but it's a little consuming to kind of think about going to a casino. You really want to put in the least amount of money to get the most money back. This right here, I have to put in a lot of money to gain a lot of money, and so that's not something my body wants to do all the time. So I can store lots of energy in these. It's exactly why if you guys don't eat for a while, like think for a day or two, you don't just die from lack of sugar, but your body is able to start breaking up all the fat inside your body, and that's where you get energy to keep going for a while. Another function of lipids, again, this is going to be compose my cell membrane. That's a huge thing, right? Because all living things start out as a cell, and I can't have a cell unless I draw that circle right there. Well, that circle is what the cell membrane is. So since it makes up my cell membrane, that's a pretty important deal. Also, we could talk about insulation and stuff like that, but really, as far as functions go, these are the 2 that you would want to focus on the most. So we covered elements, we covered the monomers and their structure, how to build them. That's where we get our triglycerides, which are the basis for most lipids. And we talked about the different structures, about how they can pack tight or not tight. And then we have the special cases, and then we have our functions. All right. Hope you guys got it.